The title of my talk is Label-Free Real-Time Monitoring of Immune Cell-Mediated Killing Using Cellular Impedance. A quick outline for my talk. I'm going to speak extensively about the xCELLigence technology, explaining how this works. And then from there, I'll give a general example showing you what the data that's generated by this technology looks like. And then collectively, these will serve as a foundation for actually looking at cancer immunotherapy applications. So very quickly, the xCELLigence technology uses microtiter plates that look very similar to any microtiter plate you've ever used before. The difference is that these plates are electronic in nature. And as you'll see over the next few slides, there are actually microelectrodes fused to the bottom of these plates. These electronic plates get placed inside the xCELLigence instruments, which sit inside a standard tissue culture incubator. So you can control the temperature, the humidity, the atmospheric composition as you would in any other assay that you run. The instrument integrates then with a control unit that sits outside the incubator. And this is where you set up, run the assay, and then analyze your data. So what I'm showing here is a blown up view of a single well in one of these microtiter plates. I'm showing it immersed in media. Again, though, the difference is that there are gold electrodes here. And so these gold electrodes along the bottom are what we use to detect the cells. When this plate is added to the xCELLigence instrument, it applies a very weak electric potential, and this causes a current flow from one electrode to the next, passing through bulk solution. This measurement here, taking place just in the presence of media alone, serves as the background. Of course, we're interested in analyzing cells, and so when you've added cells to this plate, they settle out of solution and they deposit on these electrodes. And this is a very different situation now because the cells act as insulators. That is, they reduce the flow of electric current from one electrode to the next. Another term for this resistance is impedance, and this is the basis of the xCELLigence assay. So some key features of this are that it's a very simple workflow. Couldn't be more easy. It's the same as you would in any other microtiter plate assay that you run. You use standard tissue culture media, so you don't have to use anything exotic or special here. Just go ahead with your regular experimental setup. Again, label-free conditions, so you don't have to worry about any sort of artifacts you're inducing by having to label your cells. Adhesion and growth of cells on these electrodes is absolutely normal. So we've done side-by-side assays looking at growth and adhesion on these plates versus a standard tissue culture plate like a polystyrene, and there's absolutely no difference at all. Also very important is the fact that cells are unaffected by the electric potential that they're sitting in during the course of these assays. Again, this is a real-time monitoring situation. If you'd like, you can measure your cells every 10 seconds, every 10 minutes, every 10 hours, whatever it looks like. The technology doesn't limit your assay. What limits your assay is the biology that you're trying to detect. Another important feature of xCELLigence is that it's compatible with orthogonal assays. So if you, what I'm trying to say here is that your cells are not destroyed in the process of running the assay. If you'd like to do the assay and then collect cells at the end of it and analyze your RNA, do imaging, whatever, all that's possible with this system. Finally, what I want to say is that what the system actually plots at the end of the day is a parameter that we call cell index. And this is the impedance measured at whatever time point you're interested, minus the impedance at t = 0. That is minus the background impedance prior to adding your cells to the plate. What I showed you on the previous slide is very much a simplification. In reality, what these wells look like is an interdigitation of a set of electrodes, very similar to what I'm showing here, where one electrode is shown in black and the other is shown in blue. And this is actual photo looking down into one well in a 96 well plate. You can see the gold electrode array here. The next few images just show the types of microscopy that are possible on these plates. There's nothing to limit being able to look at your cells through a microscope, either by immunofluorescence or standard phase contrast or what have you. So that's what xCELLigence is, and that's how it works. The question is, well, what kind of biology can you do with the system? Well, the first thing you can measure is changes in cell number. As you go from having a single cell to having multiple cells, the overall surface area that's being taken up by these cells now is very different. The reduction or the resistance to flow in electric current is much more dramatic here than it is in this case. Another thing you can pick up on are changes in cellular morphology. So it's amazing, but you can actually add, say, a GPCR ligand to the system, and within minutes you can see the cells begin to change shape, and you can pick it up by impedance even faster than you can by microscopy. You can also look at changes in cell surface attachment quality. So in a case where a cell is sort of loosely adherent and there aren't very many focal adhesions involved, that's very different than when a cell becomes strongly attached to the plate bottom. That affects the flow of electric current. We can pick up on this. So one of the beauties of xCELLigence is that these three parameters, cell number, morphology, and surface attachment, that encompasses a vast amount of biology. There's a huge number of biochemical pathways that impinge upon these things, especially when you consider them as a function of time. So we can look at a huge number of things. This list is not exhaustive. It's just a set of things that I thought that this particular audience might find interesting. So in particular, you could look at drug-mediated cytotoxicity, the induction of apoptosis. You can look at cell-mediated cytotoxicity. We'll talk about that in detail here today. Also looking at the inhibition of cell surface receptors, immune cell activation, cell-cell interactions. There's a huge array of things you can look at. Again, this list is not exhaustive. And when you consider the types of fields that xCELLigence is being used in, it's everything from cancer and inflammation to regenerative medicine, toxicology, virology. The list is quite extensive. At this point, we have about 1,700 of these instruments placed globally. And as of last week, we're around 1,250 publications, so it's very well adopted at this point. What I'd like to do now is show you a very general example, and I'm choosing this example of cellular adhesion because I think it nicely demonstrates the correlation between the impedance signal that xCELLigence measures and actually what's going on with your cells as detected using an orthogonal technique like microscopy. This is a very simple experiment. We've added the lung cancer cell line A549 cells to the wells of the plate, 5,000 cells per well. The only difference is that the wells have been coated with different concentrations of collagen. You don't need to use a coating matrix or a coating protein when you do the xCELLigence assay, but for some cell types, obviously that's useful. And so we're trying to look here at how that impacts how quickly and how tightly the cells adhere to the plate bottom. What you can see here is that there's a very distinct difference. In the absence of any collagen, the impedance signal is quite weak, but there's this really nice correlation between the concentration of the collagen that's been used and the impedance signal. And what's really beautiful about this is that you can take the plate over to the microscope and see a perfect correlation between the impedance signal and actually the number of cells that are attached to the plate. So in the presence of no collagen at all, you see very few cells attached, and the cells that are attached are very loosely adherent. They're very spherical and they haven't laid down onto the plate bottom. In contrast, at higher collagen concentrations, you have more cells, and the cells have attached more tightly. So I want to move on now to actually look at cancer immunotherapy applications. And so I'm going to provide you with sort of a general overview of what this type of an assay looks like using xCELLigence. The first thing is that you need to add I'm going to address adherent cell lines first. I'll address liquid non-adherent cells in a minute. You would add your adherent cells to the plate, and there's an expectation that these cells are going to proliferate, obviously. At some point, the cells have become confluent and they've taken up all the real estate on that plate bottom, and so the impedance signal will plateau. What's really beautiful about this assay, though, is that the effector cells that you would want to study are not naturally adherent, and because of that, they interact very weakly with the plate bottom. And because of this, we expect the effector cells to have very minimal signal on their own. And the benefit of this assay is that this allows for us to, in this system, look at what's happening to the target cells by themselves. The signal from the effector cells is basically non-existent, and so we're asking, how do these effector cells interact with the target cells, induce apoptosis, morphological change, and then detachment from the plate bottom? And so what we would expect is in the absence of effector cells, again, you would see a plateau. But as you titrate in more and more effector cells, you would expect for the response to be more robust, not only in the rate at which killing is occurring, but also the extent of killing that's taking place. And you can imagine there's all sorts of things that you could evaluate using this system. Serial killing, how persistent a given type of effector cell is. If you're familiar with these assays, a common assay technique is to label cells with chromium-51 and then look at the release of chromium-51 as the target cells are lysed. This gets rid of that situation. You can do an assay anywhere from minutes to days. Again, the technology doesn't limit the assay, it's the biology that limits the assay. And then there's a big value in being able to track these systems continuously rather than having endpoints which are just snapshots over the spectrum of what's taking place. So the first example I'll look at is natural killer cells mediating cytotoxicity. This is a very simple example of MCF7 target cells that are being treated with NK92 cells. And you see here a really nice response. What we have in blue are the untreated cells. You see them continuing to proliferate. They haven't reached confluence yet, and so the signal is not plateaued. But as you titrate in greater numbers of effector cells, there's obviously a more robust response. So this looks just like the theoretical plots I showed you on the previous slide. I want to highlight that there's a very high sensitivity even at low effector to target ratio. So you can see here at effector to target ratio of 0.5 to 1, we're still picking up on killing. That type of sensitivity is very hard to achieve, even impossible, using traditional cell killing assays, something like a chromium-51 release. You can rapidly detect the killing, so within about 2 hours, you're already seeing a response. And so a good question would be, well, how do you know, okay, we see a change in impedance, how do you know that's actually cell killing? Well, it's a very simple thing to go ahead and then go do an orthogonal assay. Whether you want to do a Western blot or immunofluorescence or what have you, you can verify that this drop in impedance signal is actually, is in fact apoptosis and killing of the target cells. Again, the technology doesn't ruin the cells in the process, so it's very easy to use an orthogonal approach. The next example I'm going to give you is that from looking at the killing of liquid tumor cells. So obviously a huge thrust in immunotherapy right now is focused on being able to kill liquid cancers. The system I've described to you is tailored towards looking at target cells which are naturally adherent. Over the last year, we've developed an assay, an actual kit that allows for us to look at the killing of liquid tumor cells. In particular B-cell cancers. And so the way that this works is that we coat the electrodes with an anti-CD40 antibody, and then this allows for us to tether B-cells that are expressing the CD40 protein on their surface. You can then add on top of this the effector cells and monitor the killing process. What I'm showing here is a set of controls. In red are Dowdy, which is a B-cell line. After they've been added to a plate that has antibody, the CD40 antibody already on its surface, you can see the attachment and growth to the point of confluence, as you would expect. But when these plates don't have antibody on the surface, you see that there's very little signal here, as shown in green. That's exactly what we would expect. Importantly, what I'm showing here in the blue and purple are NK92 cells that have been added either with or without the antibody coating. Again, this is what we would expect. There's very little signal from these because these effector cells are not naturally adherent. They're not interacting with the electrodes. It's a beautiful system. It allows us to look only at the target cells. What I'm showing here is the actual data. In green, you have the untreated sample. And then again, as you add in more effector cells, you see this, this nice dose-dependent response. So exactly what we'd expect to see. This allows for you to evaluate a huge number of things. And I'll go into some more details here over the next few slides about the different types of variations in this assay that are possible. I want to say that at the moment we've had success looking at a variety of B-cell lines with this, but we're also working on being able to look at primary B-cells. We'd love to be able to isolate blood from a patient and then look directly at their cancerous B-cells as a way of being able to find the most effective treatment prior to actually administering it. So an important question would be, well, you've now tethered these target cells to the plate bottom. How does that impact the biology? And I'll very quickly say that we've done side-by-side assays where we've done the xCELLigence assay and then done the in-solution killing assay using the Novocyte flow cytometer, just tethering versus in solution. And what I'm showing here is the percent cytolysis as a function of different effector to target ratios. And you can see in blue we have xCELLigence and in red we have flow. The systems are basically identical, okay? This is evaluated at 4 hours. When you evaluate at 24 hours, you also see that there's a very strong correlation. They look very similar. So at least in this context, we've convinced ourselves that we're not perturbing the biology. There may be other cases where that is not the case, but I'll tell you that a goal of ours is to develop multiple kits along this line that will allow us to look at different types of cancer target cells. I'm going to go ahead now and move on to an example of T-cell mediated cytotoxicity. And I'm actually going to, for the sake of time, jump through this and move to an example using chimeric antigen receptor T-cells. Okay, so most of you are probably familiar, but in this case, you're assembling a chimeric protein, the extracellular domain of which has been engineered to bind to the antigen of interest on whatever cancer cell it is you're looking at. And then the intracellular domains involve signaling for activation of those T cells. So a common problem with this, although this technique can be very effective, one of the challenges is that not every single cancer cell is identical. It's a heterogeneous population, and the antigen that you have engineered your CAR T cell to bind to may not exist on every cancer cell in that particular tumor. And so in this case, I'm just showing generically this green chimeric antigen receptor. It'll take care of all these cells which are expressing that antigen, but the problem is there may be 1 cell or 10 cells in that population that don't express that antigen. These are the cells that persist and then go on to seed tumor growth later on. And so there's an interest in this field of designing chimeric antigen receptor T cells that target not one but multiple antigens simultaneously. And the goal here would be that you cast a broader net and you don't allow for these escaping cells to then seed subsequent tumor growth. And so in this particular example, you might talk about adding 2 of these CAR molecules per T cell. This example is from a group at Baylor College of Medicine. They were trying to target the HER2 protein and also IL-13R alpha 2 at the same time. And what was very interesting is they said that there's multiple ways that you could go about trying to target both of these antigens. In one case, you could put 2 different populations of T cells, each of them expressing 1 CAR. As an alternative, you might put both of those CAR molecules into the same T cell. And finally, something they did that which is very creative is that they fused both of the CAR molecules into a single protein and put that into a T cell. And they asked, well, what is the difference in killing efficacy between these different scenarios? This is the data. I won't go into every single trace here, but I want to highlight the fact that what they found is that this tan CAR situation where they fused both molecules together, shown in red here, that ended up being the most effective cell killer. And the data that they found using xCELLigence correlated perfectly when they took these constructs into mice. The killing efficacy by xCELLigence matched what they saw in vivo. So these types of assays where you have a continuous data trace over multiple days enable you to look at subtleties in cell killing that you're not going to be able to pick up on using a traditional endpoint assay. I don't have time to go into a great detail on 2 of these examples, but I will go ahead and mention the case of BiTEs. So if you're not familiar, BiTEs are bispecific T cell engagers. These molecules have been designed to bind to CD3 on the T cell and then bind to whatever antigen it is that you're interested in on the target cell. And the whole point is that by bringing these into close spatial proximity, you're expecting to activate the T cell and induce apoptosis in the target cells. This particular example is from Genentech, Judy Young's lab, and so I can't share details about what tumor cells they looked at or what antigen they looked at. But I can just say that the T cell to tumor cell ratio is 4 to 1. I'm going to move through this fairly quickly here. What you can see here in the case of using a cell line that's not expressing the antigen of interest, you don't see any response at all. This is just sort of a standard proliferation. However, when the cell line expresses the antigen of interest that the BiTE molecule is specific for, you see a very distinct concentration-dependent response. So we're looking at 0 up to 100 nanograms per mil of the BiTE molecule. They did this for multiple clones, and you can see they're able to tease apart differences. Clone 2 looks very similar to clone 1. However, clone 3 here, the response is not as robust, and you begin to see some of these cells, although there is some killing, they begin to recover. These are the types of changes that you can pick up on using this technology. It's very simple to go ahead and try and correlate the results from xCELLigence using orthogonal assays. And I'll just say very quickly that when you look at the data collected by xCELLigence to try and get, say, an EC50 for the BiTE molecule, the values were between 0.75 and 1 nanograms per mil. If you look at it by flow cytometry, they're very similar, okay? And this is a very common theme that we've seen throughout our work in our own labs, but also in the literature, where the data with xCELLigence correlates very well with orthogonal approaches. So it gives us a lot of confidence that these types of real-time assays are in fact, giving you data that's trustworthy and that's physiologically relevant. So at this point, I'm going to go ahead and wrap up. I want to say this though, if you're going to use a traditional cell-killing assay, one approach would be to monitor the effector cells. You might look at perforin or granzyme secretion, you might look at cytokine production or different signaling pathways, but something that you need to be aware of and reconciled with is the fact that these different biochemical reductionistic approaches very often do not even correlate with the efficacy of how well these cells kill target cells. That's something that's been borne out over and over again in the literature. So I'm not saying that those biochemical approaches are not useful, but I'm just saying that if the end goal is to find a treatment that's the most effective killer, that's not your best approach. You can also monitor the target cells using a release assay like chromium or fluorescent dye, or the release of an endogenous biomolecule. The problem with these approaches is that you're using radioactivity. There's a very short assay window. If you're using chromium, your assay window is about 4 hours. I just showed you examples with xCELLigence where the assay window is 140 hours without any problem at all. Other issues, if you're using the approach of monitoring the target cells, are low sensitivity and poor signal to noise. So in conclusion, I hope that I've shown you that xCELLigence has a very simple workflow, that it allows you to do assays in a label-free manner. It's non-invasive, so you can use your cells for any other approach you'd like during the course of the xCELLigence assay. And it's widely applicable, anything from virology to toxicology to cardiac toxicity. And I think that xCELLigence gives you a very complete picture of cell health and behavior as long as you're using the appropriate controls in conjunction with this. So with that, I'd like to thank my colleagues, Fabio and Biao, who have done a lot of the work in the immunotherapy area. A lot of the data I've shown is directly from those guys. I'd be happy to take any questions if you have any at this point.